Right, so last time we were talking about the white blood cells, right? We have discussed into detail your friend neutrophil, how it works, right? Today we will talk about the remaining two granulocytes, which are the functions of eosinophils and basophils. Let's first of all talk about the eosinophils, right? Eosinophils, already you know, are the white cells which have granules, specific granules of which color? Yeah. The very special thing about the eosinophil is that they have bilobed nucleus, right? I always feel somehow eosinophils are having some sort of glasses, I think, right? They are looking around, and eosinophils have bilobed nucleus, and they are having specific granules, and these granules are taking which color, please?
Red.
Red color. And but what we call the red color? Acidic color, or eosin simply, right? So acidophilic granules, because they love the acidic colors, or acidophilic granules. So eosinophils are the white blood cells which have large granules which take the eosinophilic dyes or acidic dyes, right? And eosinophil normal number is somewhere between 1 to 6% of all white blood cells. Is that right? And about the granules, what are the different substances present in these granules, the eosinophilic granules, right? What type of substances they have in? Yes, who will tell me? Number one, these granules are very rich in, yes, eosinophilic, yes, eosinophilic basic proteins, right? Eosinophilic basic proteins, number one. Number two, they are having eosinophilic cationic protein. Eosinophilic cationic proteins. Number three, these granules are having eosinophilic peroxidases. And these three products are very dangerous for parasites and protozoan, right? So protozoans and parasites, these products are cytotoxic. Cytotoxic means they kill the cells. Cytotoxic to protozoans and helminths, right? Now you can understand that eosinophils play a major role in killing protozoans and helminths, right? And anyone who is heavily infested with the parasites in the blood of this person, eosinophil number will go up, right? And if you are heavily loaded with parasites or protozoans, then your eosinophil number will be more than 6%. And if your eosinophil number is more than 6%, we say in the blood you have eosinophilia, right? Then another special product which is present in these granules, specific granule, is eosinophils derived neurotoxin. That is eosinophil derived neurotoxins. This substance can produce a dysfunction in Helmint's nervous system, but not protozoan, because helminths have, these are multicellular parasites, they have nervous system, right? And eosinophils produce special neurotoxins which paralyze or produce dysfunction in helminth's nervous system, right? Then something very interesting, that eosinophils have special type of two product. Okay, let me draw a eosinophil for you, right? What is the shape of the nucleus? Bilobed. Is that right? Number one. And what is the color taken by the granules? red, right? Red color is given by which type of dye? Eosin, which is basically acidic dye, right? Now, another thing, okay, let's make it a happy eosinophil. Right? It's a happy eosinophil because you are learning about it. Right now it's center of attention. Fine. Now, these eosinophils, they have two more special products, right? And those special products, yeah, what are those special products by eosinophils? You have to remember them. Who will tell me? Those special two products, they help the eosinophil to relax. Yes, to reduce the inflammatory response. Actually, eosinophils have a capability to produce histaminases. I think they produce like this. Histaminases. Histaminase is a group of enzymes which can break down. Yes, please. They can break down histamine, right? And if the eosinophils are producing the product like histaminases, which can break down the histamine, right, then it means that the focus of injury histamine level will go down. And histamine mediate many steps of inflammation. Histamine is vasodilator. Histamine activate the endothelial cell, right? It make the endothelial cell more adhesive to the white cells. So because histamine produced by the mast cells classically enhances the inflammation, and your friend eosinophils produces histaminases, so it means it breaks down the histamine. It means it increases the inflammation or decreases the inflammation? It decreases the inflammation. That's very good. So it decreases. So you can say when eosinophils infiltrate into tissue, they release their histaminases, and in inflammatory area, histamine's concentration go down and inflammation is toned down or reduced. Secondly, they produce one more substance, and that is called aryl sulfatases. Aryl sulfatases. Aryl sulfatases actually break down leukotrienes. They break down leukotrienes, and histaminases break down histamine. Right? So it means the very important two functions that it aryl sulfatases break down leukotrienes. Leukotrienes are the product produced at the site of injury, right? And especially leukotriene B4 is producing attraction for the neutrophils. Leukotrienes, I think spellings should be, in the books it is written leukotrienes, but I think it should be leukotrienes because these substances produce the trains of the leukocyte. When you put in any tissue some leukotrienes, then from the blood vessels, nearby blood vessels, leukocytes come out and like trains, they start moving towards the leukotrienes. Is that right? So leukotrienes have two important functions. Number one, they are chemotactic agent for neutrophils, especially leukotriene B4, and there are leukotriene C4 and D4 which activate the endothelial cells and produce spasticity of bronchial smooth muscle. Leukotrienes are produced in high concentration in allergic reactions because histamines and leukotrienes are product of the mast cells which are activated during allergic reactions, right? But once eosinophils come, through histaminase release, they destroy the histamine, and through aryl sulfatases release, they destroy the leukotrienes. In this way, when histamine is destroyed, many vascular events are reduced in the inflammatory side, and when leukotrienes are destroyed, endothelial activation is reduced as well as leukocyte infiltration into tissue is reduced. Am I clear? Any problem up to this? There's really no problem? Right. Then, okay. Yeah. This eosinophil, it is having some I think, napkin in its hand to remind you something. Right? Napkin. N-A-A-P-C-A-N. I think Sinophil doesn't know what are the proper spellings of napkin. Is that right? So, Sinophil having napkin, right? And dancing around with red face and black googles. Is that right? And having special type of ears, right? And 2 napkins. Why putting 2 napkins? To remind you something about the napkins. Yeah, napkin. N-A-A, yes, P-C-A-N. Actually, N stands for neoplasia. In some neoplasia, eosinophils concentration in the blood increases. Actually, napkin should remind you what are the circumstances in which eosinophilia is produced. What are the situations or pathological situations in which in your body eosinophil level goes up, right? Now, N stands for neoplasia, A for allergy. In many allergic reactions, especially type 1 hypersensitivity reactions, eosinophils go up. Then another A, especially asthma. Of course, it's one of maybe due to allergy. So some patients with special type of neoplasia, they have eosinophilia. Some people with allergic reactions have eosinophilia. Some people with asthma have eosinophilia. And P should remind you. Yes, P should remind you. Parasites. That's good. So parasitic infestation, parasites. That also produces what? Eosinophilia. And C should remind you collagen vascular diseases, which you will study in pathology. Vascular diseases. These are special type of autoimmune diseases, and some of the collagen vascular diseases have eosinophilia in the patient. Right? After that, what is there? Neoplasia, allergic reactions, asthma, parasites, collagen vascular diseases, and A for what? And N for what? For repetition. A again for allergy to remind you. And N again for neoplasia. So you have to remember it. That these are 2 very important situations in which eosinophilia is there. Right? If you forget that, just remember that eosinophils try to remind you by napkins. And then you must know that spellings of the napkins, eosinophil is a bit weird about it. Is that right? Am I clear? So again, let's revise it. Eosinophils are granulocyte with what type of granules? Eosinophilic granules. What type of nucleus? Bilobed nucleus. And neutrophil were having what type of nucleus? Multi-lobated nucleus, maybe 2 or 3 or 4 or even 5 lobes of the nucleus in case of neutrophil, right? So we can compare also okay, these are 5 white cells. Let's differentiate them with the nuclei. Neutrophil have multi-lobated nucleus, is that right? Eosinophil has classically bilobed nucleus. And what is it? Basophils have S-shaped nucleus. And then when we come to kidney-shaped nucleus of monocytes and very big nucleus of lymphocytes. And some lymphocytes can convert into a cell which has a cartwheel-shaped nucleus. What is this cell? Plasma cell, which is an active B cell producing antibodies. Right? So it's clear how you differentiate the white cells just by nuclear morphology. But of course, more wise that you try to differentiate them with nuclear morphology plus with their granules. The neutrophils have granules, specific and non-specific both. Remember, all of the white cells have non-specific granules, lysosomes. But specific granules are only in granulocyte. Non-specific granules are present in all of them. And here the specific granule here are in neutrophil. What stain is taken by the granules? Some take blue color, some take red color. And in case of eosinophils, Granules take red color. In case of basophils, they take blue color. Basic dyes, right? And then very important function of eosinophil is that it reduces inflammatory reaction in the tissues where they go by producing histaminases and aryl sulfatases. Histaminases reduces the histamine concentration there. Aryl sulfatases reduces the leukotrienes there, right? Moreover, under what circumstances eosinophils are very high in the blood? That is neoplastic, some of the neoplasias, type 1 type of allergies, asthma, parasitic infestations, collagen vascular diseases, and just to stress again, allergic and neoplastic conditions. Any question up to there? There's no question. Fine. Now we come to the last granulocyte which we are supposed to discuss, and that is, yes, basophils, right? Yes. What is your question?
Does the eosinophil and basophil go through the same 5 stages of?
Okay, there's a very important question. She remember that statement, right? She's saying that as when neutrophil develops, during the development of the neutrophil, there were different stages. If you remember from last video, that neutrophil development requires the special stages of, what was first? Myeloblast, then promyelocyte, then myelocyte, metamyelocyte, stab cells, and eventually mature form of neutrophils. Actually, this is called stages of granulopoiesis. Actually, development of eosinophils and basophils also require the similar stages, right? Except that usually in case of basophils and development of the eosinophils, after the metamyelocyte there was a stage of stab cell, right? Probably stab cell stage is not that clear, right? And metamyelocyte stage is not clear. But again, you have to remember that almost similar stages are seen during the formation of eosinophils and basophils. Is that right? No problem up to this? Except that all these 3 granulocytes have different type of specific granules. Is it clear? Okay. Now we come to the basophils, right? So let's talk about basophil. This is your friend, basophil, and its nucleus is, it is bilobed, but it is more like S-shaped. I think it's more stylish, isn't it? It's more stylish as compared to eosinophils. And it is having, what type of granules? Right? Basophilic granules. Is that right? And I think I'm too stylish, isn't it? So this is your basophil, right? And important thing, they are having a beautiful crown also here. What are this crown? They are having receptors on which some antibodies are loaded. These are special receptors on basophils on which the immunoglobulin of E class, IgE, that is loaded. So you can say basophils are circulating in the blood, and on the cell membrane of the basophils there are receptors for IgE. So basophils are coated with immunoglobulin E. Is that right? Which plays a role in type 1 hypersensitivity reaction, right? So this is about basophils, and basophils are having, of course, some products. I think they are too stylish, isn't it? Right. We'll talk about this tail, why it's so beautiful tail. But one thing before, what is present in their granules? What comes out of their granules? Yeah, who will tell me what is coming out of these granules? Basophils granules, they release heparin, they release, yes, heparin, Heparin acts as an anticoagulant, right? With heparin, they also produce a similar molecule called heparan sulfate. Heparan sulfate molecule. Heparin and heparan, they act as, yes, anticoagulant. They also produce histamine. Histamine is very important chemical mediator which is produced during type 1 hypersensitivity reactions, special allergic reactions, right? And they also produce what? Leukotrienes. Leukotrienes are also very powerful chemical mediator for inducing inflammation. Is that right? I told you leukotrienes are playing a role in endothelial activation. Leukotriene also produce a role in producing chemotactic effect on the neutrophils, right? Now, here it's very important to remember that your eosinophils were producing histaminase, but these are producing histamine, right? So it means eosinophil destroy the histamine, but basophils produce histamine. Another thing that eosinophils were producing aryl sulfatases, which were destroying leukotrienes, but actually your friends, stylish friends, basophils produce leukotrienes. Is that right? And basophil are really, stylish people are not common, including these. They are very rarely found in the blood, 0 to 2%. Actually, to find a basophil, sometimes you have to study many, you can say, slides of the peripheral blood picture to find some basophils, right? They are not very commonly present. So sometimes in some slides you may not see any basophils, right? And maximum you are allowed to find up to 2. But if you find more than 2% basophils, right? 2% of all white blood cells, we say there is basophilia. Is that right? There is basophilia, right? Another thing, under some circumstances, eosinophils becomes, what is this? Stippled. What is stippling? Yeah. Eosinophils look stippled. Their cytoplasm looks having lot of dots. Stippling. Stippled basophils. Stippled basophils. Right? So for that stippling, you have to remember what thing? What is this? Tail. So you have to remember the tail. Tail stands for what? T for thalassemias. Thalassemias are a special type of anemias, and in thalassemias, basophils become stippled. Right? T for thalassemias, A for, yes, T for thalassemias, A for anemias of chronic diseases. Anemias of chronic diseases. For example, someone has anemia due to rheumatoid arthritis or anemia due to tuberculosis, right? In such patients, basophils may be stippled. Then TAI, I for iron. Iron deficiency, anemia, right? And in the end there is L, and L stands for lead poisoning. Lead poisoning. If someone has lead poisoning, eosinophils will become stippled. How you get the lead poisoning?
Paint.
Yeah, paints. Some paints are having lead, and of course some children, they like to eat the paint, right? Somewhere they find in the home some place like basement, and they go there and every day they enjoy some paint and then clean and then come up, and they always get surprised how mother knows it because there's a big paint here, right? So some children who take a lot of paint regularly on long-term basis, they get lead poisoning, which develops many features, including this stippled situation on the eosinophils. Is that right? So what are the important point about eosinophils? Number one, are they, oh yeah, sorry, basophils, not eosinophils, basophils, yes, thank you for correcting it, right? Number one, that basophils, what is the percentage of basophils in the blood? 0 to 2%, right? Thank you for looking. Secondly, yeah, I should appreciate that. Right, you use both eyes. Now, number one. Number two, eosinophils. What are the basophils? What are the granules color? Blue. B for blue, B for basophils, right? B for basic dye, right? All of them with B. Then basophils, they become what? Stippled. They develop stippled basophils in the blood. Under what circumstances? In thalassemias, anemia of chronic diseases, iron deficiency anemia. Yeah, what is this? And lead poisoning, right? So these are a few words about your friend, stylish friend. And now we have finished the neutrophils and eosinophils as well as basophils, right? Yes. Stippled. Stippled means that any cell which is having multiple dots. Is that right? We say it's stippled. Do you have any other meaning of stippling? I know only one meaning, that if there's any cell and it's having multiple dots, right? We say stippled, right? Okay, so now we come to which cells? Monocytes, right? What we have done up to now. You remember, just to remind you, we are studying white blood cells, and we have divided them into granulocyte and yes, please. No, agranulocyte or non-granulocyte, right? Granulocyte were divided into, what were these friends? Neutrophils. And what is this place? Eosinophils. And what is this place? Basophil, right? And non-granulocyte are divided into two. They are, yes, kidney-shaped nucleus monocytes, and what is this? Lymphocytes. So we have discussed them. Now let's come there about the monocytes. Monocytes are also produced in the bone marrow. Is that right? And their percentage in the blood is somewhere between 2 to 10%. I have previously explained that monocytosis will occur when the percentage of monocytes out of all the white cells in the blood is more than 10%. And monocytosis occur especially in chronic inflammations, chronic infections or chronic inflammations, monocytosis. Okay. When monocytes come from the bone marrow house, of course, first they jump into where? General circulation, right? So they come into general circulation. In general circulation, monocytes survive about 2 to 3 days, and after that they may shift to the tissues. Is that right? And in their tissues, monocytes are called macrophages, and there they live for years and years sometimes. They have long life in the tissues, monocytes, right? Now, once monocytes go to the tissues, in some tissues, monocytes are present all the time. Even if the tissues are not inflamed, monocytes are present there, right? And there are other tissues in which monocytes go only when there is injury and inflammation, right? Now, the tissues in which monocytes normally go and take a permanent residence are, number one, central nervous system. Monocytes go to the central nervous system and live there for long time. They convert into special type of connective tissue cells, right? Actually, monocytes go to the developing neural tube, and when out of neural tube central nervous system is developed, those monocytes which had migrated to the neural tube, they convert into very special type of cells into central nervous system. What are those cells called? They are called microglia. Microglia, right? So microglia in the central nervous system are just monocytes which are taking permanent residence there. Then monocytes may go to, what is this tissue? Yeah, lungs. Thank you for recognizing it. So sometimes in the alveoli, rather always in the alveoli, monocytes or macrophages are patrolling in the alveoli and looking for some particulate matter to phagocytose, right? And these are called alveolar macrophages. They are normally present in your air spaces in the lungs or in the alveoli. Then some of the monocytes take permanent residence in liver, and there these monocytes are called, Kupffer cell. Kupffer cells. Have you heard of Kupffer cells? These are basically macro monocytes which have gone. Actually, once the monocytes are out of the circulation, they are called macrophages. So we should say that the macrophages in central nervous system are microglia, macrophages in the alveoli are called alveolar macrophages, macrophages in the liver are called Kupffer cells. Then macrophages, if they go into, yes, lymph node or into, yes, spleen. So what they are called? They are called histiocytes. Actually, macrophages which are present in all the tissues are called histiocyte. Histio means tissue, cyte means cell. So macrophages which are present in almost all tissues, they are called histiocytes, right? But for example, some of the macrophages are present in the lymph node sinuses. They are called sinus macrophages. They are called sinus macrophages. In the same way, you must remember that very big number of monocytes or macrophages are present in spleen, which are concerned with the phagocytosis of old RBCs. You know that, especially in cords of Billroth, right? So in the spleen also lot of macrophages are present over there normally. Then macrophage love to shift to some other tissues, like if they shift under the skin, in the dermis. What are they called? Langerhans cells. Langer, L-A-N-G-E-R, Langerhans cells. Then macrophage love to shift to the kidney, and in the kidney they are called mesangial cells. Mesangial, yes, cells. And then macrophages love to shift to the, you have to recognize this tissue. Here they are called osteoclast. What are they called? Osteoclast. Osteoclasts in the bones are very special type of macrophages, right? Then can you tell me macrophages in some other part? And of course, macrophages may go to the tissue when tissue is inflamed. When there is inflammation, macrophages are second to neutrophil to appear into inflamed tissue. So they are cells of neutrophil and macrophages. This combination is cells of acute inflammation. But if macrophages are with the lymphocytes and no neutrophil, then these are the cells of chronic inflammation, right? So during inflammation also, monocytes shift to the tissues, right? Additional monocytes converting into macrophages in the tissues, and what they are doing in the tissues, right? What monocytes are doing in the tissues? They have three important functions. Number one, they are phagocytic cells. They are involved in phagocytosis. Normally macrophages phagocytose old RBCs, old platelets, even old other white cells. Then macrophages also phagocytose bacteria or other particulate matter. Monocytes also phagocytose our injured cells or injured cells debris, right? For example, if there's some injury and many of your cells die, macrophage will come into tissue and remove the dead cell so that injured tissue get rid of the dead cells, and then local cells proliferate and replace the injured tissue, right? Lost tissue. So what is really happening? One of the function of macrophages is that macrophages are very powerful phagocytic cells. Then another very important function of the macrophages is they are very, very important antigen presenting cells. You must be knowing that macrophage engulfs some antigens, circulating antigens, process the antigens with class 2 or class 1 molecules and present to the T helper cells. Is that right? Here I must remind that all the cells in your body are having the capability to express class 1 molecules, except RBCs. But only antigen presenting cells have capability to express class 1 plus class 2 molecules. And what are the antigen presenting cells? Number 1, macrophages. Number 2, B lymphocytes also act as antigen presenting cells, B lymphocytes. Number 3, dendritic cells, especially follicular dendritic cells. Number 4, all the derivatives of macrophages, like Langerhans cells and others. They have a capability to process the antigen and present the antigen with class 2 molecules. Is that right? Any question? So, and of course, you have to tell me that do macrophages have any granules or no? Yes. They do have granules, non-specific granules, right? Which are having xerophilic granules, also they called non-specific granules or xerophilic granules, the primary granules, right? Or simply call them lysosomes. These are all one and the same type of granules. So these granules are present in monocytes as well. Is that right? Do you have any question? No. Now we come to the lymphocytes, and we have discussed lymphocytes in a big detail when we study the immunology, but just let me tell you something about the lymphocytes as a revision only. Right? You will study lymphocytes in very big detail in the lectures of immune system or lymphoid system, right? But just few words that there are lymphocytes are basically how many types of lymphocytes? Yes? Classically, there are 3 types of lymphocytes. There are B lymphocytes, and there are T lymphocytes, and there are natural killer cells. Remember, B and T lymphocytes are part of the adaptive immune system, but natural killer cells are the part of innate immune system, right? And just to remind that neutrophil, eosinophils, basophils, and natural killer cells and monocyte macrophage system, all of them are part of innate immune system. Is that right? Now, B lymphocytes and T lymphocytes. T lymphocytes are classically divided into how many types? 3 types. T lymphocytes may be T helper cells, very good, T cytotoxic cells, and T regulatory cells, right? Or T suppressor cells. All the T cells, then T helper cells are divided into 2 types of cells. Yes, please. T helper 1 and T helper 2 cells. Then you must be knowing T helper 2 can produce cytokines which can activate B cells and convert them into, yes, plasma cells, and plasma cells are producing antibodies. And T helper 1 classically help the, yes, macrophages, right? T helper 1 classically help the macrophages to convert into superactivated macrophages or into epithelioid cells or giant cells, right? So you can say T helper 1 activate the macrophages, right? This is because T helper 1 produce a product like, yes, T helper 1 produce a product like tumor necrotic factor and gamma interferon, right, which activate the macrophages. And T helper 2 produce product like interleukin 4 and interleukin 5, which specially help the B cell to convert into plasma cell. But T helper 1 and T helper 2 both produce interleukin 2 for their stimulation, right? So we have already discussed them into detail, right? There's no need to discuss it here, and that's all about the white cells.